So thank you for the invitation to speak, and thanks to all of you for coming along tonight. I appreciate it and hope there'll be something of interest to all of you. And as you've heard, I'll be more than happy to answer any of your questions afterwards. So I'm going to talk to you about how the brain changes. You'll all be aware that during development in children, there's huge changes that occur in the brain as babies learn new skills and learn to behave within the world. But what my group are particularly interested in is how the adult brain continues to change throughout life. And here are some illustrations of the kinds of things that we're interested in studying, the kinds of experiences or activities or types of learning that might shape the brain even in the adult. So for example, if you take up a new skill such as learning to play the piano, that will change your brain. If you start a new sport like juggling, again, that will change the way that your brain represents movements. The job that you do day to day will have had its effect on your brain, and you'll hear in a moment about taxi drivers. Similarly, your lifestyle, how physically fit you are or how much you engage in thinking activities in your day-to-day life, all of that will influence the way that your brain is wired up. And then also if your brain suffers any kind of injury, such as a stroke, that will affect the way it behaves, and the intact bits of your brain will be able to respond and to some degree to compensate for that damage. And I'll be telling you about some of that work also later on. So just to give you an overview of the kinds of things I'll talk about, I'll first of all describe to you some of the methods that we use to study remodeling in the adult brain, and then give you some examples of how experiences or learning can shape the healthy adult brain. Then talk about brain injury, in particular stroke, and how we might be able to stimulate or accelerate recovery after stroke. And finally, talk to you about a few new studies aiming to slow cognitive decline and dementia with increasing age. So the method that we are typically using to study brain remodeling is brain imaging, and there's a type of imaging we use called magnetic resonance imaging, or MRI. I'm sure many of you are familiar with the term MRI. It's the same scanners that you would use, say, to scan a knee injury or a shoulder injury that you have in all of the hospitals. We're using these same machines to study the brain. And we can use this machine in different kinds of ways to tell us different types of information about the brain, to tell us about the structure of the brain, the physical properties of the brain, or to tell us about how the brain's working. So structural MRI, for example, will be a bit like taking a picture of the brain. It'll tell us what the brain looks like. It'll allow us to see inside the person's head, to have a look and see what their brain looks like. There are different types of structural MRI which give us different information about the tissue properties. So here are just a few different types of pictures that we might take with our MRI scanner, and you can see that the colours are quite different across them, and depending on exactly what features we're interested in looking at, we would set up the scanner in a slightly different way. We're also interested in measuring brain function, and we can use that same piece of machinery to measure the brain at work, so to try and see what activity is going on while you're lying in that brain scanner. So here's some examples of the kinds of activities that we might see in Homer Simpson's brain, for those of you who are familiar with that character. But in most people we might see a different pattern, and we would expect to see similar sorts of responses across different people. So the way that we would set up our experiment to study brain activity would be to put our volunteer into the scanner and then ask them to do something while they're lying in the scanner. So they don't just lie still, they follow some instructions given by the experimenter, so that we can see how their brain responds to that activity. So in this very simple example, we might have a volunteer lying in a scanner, and we instruct them to move their hand every 30 seconds or so. So they alternate between periods when they're lying at rest and periods where they're moving their hand, and they do that again and again over the course of, say, 5 minutes. And we're continually taking pictures of their brain every few seconds, so that then for every location in the brain, we can look at how the picture varies over time, looking for areas in the brain when the activity that we record goes up and down together with the movements that they're making. So areas in the brain which are quiet when the person is lying still, but which are active when the person's moving their hand. And we can look at those areas like a picture of the brain. So here you see a picture as if I've taken a slice through somebody's head horizontally like this, and this is the cross-section through their brain where you see the different brain areas, and those red areas which are lit up on that brain picture, they show us areas which were active, which were working hard when the person was moving their hand compared to when they were lying at rest. So we can use this technique to pinpoint the job that different brain regions do and to pinpoint how active different brain areas are when we ask people to perform different tasks. And that's not just restricted to movement. So there's all kinds of different things that we can ask people to do in this scanner. In the example that you see here on the left-hand side, you see the kinds of activity that we'd see when someone was moving their hand, like I just showed you. In the middle, you see activity that would be generated by somebody speaking. So we there expect to see activity in the left-hand side of the brain, in particular areas near the front of the brain. And then on the right-hand side, you see in blue areas that would be active when someone was hearing sounds, and in red to yellow areas that would be active when someone was seeing pictures. So you can see then that we can use this technique to map out the different jobs that all of these different areas of the brain are doing, and then see how that activity changes either when we learn new things or when we recover from brain injury. And then the final type of technique that I'll be talking to you about today is again using that same MRI scanner, but we set up the machine in a slightly different way, now allowing us to measure characteristics of the fiber pathways of the brain. So the connections between different brain areas, what's sometimes referred to as white matter. So when people talk about grey matter, they're talking about the bits of the brain where all of the cell bodies are, the processing units of the brain. When they talk about white matter, that's the connecting pathways between those different brain areas, the pathways that communicate messages between different bits of the brain. Here you see a picture of a real human brain at post-mortem, so the top of the brain has been taken away to reveal these fibers, these underlying fiber pathways. So these are literally like little wires that will communicate electrical signals from one part of the brain to another, allowing all these different brain regions to talk to each other when we behave. So we can, the reason why we can be sensitive to these properties with our brain scanner is that we can set up our scanner to be sensitive to diffusion of water molecules. And we know that if you are in a bit of brain tissue with highly structured organisation, like a fibre pathway where we have all of these fibres aligned parallel with each other, we know that those tiny, tiny water molecules will diffuse more easily along the axis of those fibre bundles than they will across the axis of the fibre bundles. So what we do is take lots and lots of different images, and then we do all kinds of fancy computing on them in order to figure out what the orientation of those fibre bundles are and how strong those brain connections are, how strong the wires are between different brain areas. So we have these three different techniques. Structural MRI allows us to take a picture and look at the physical properties of the brain. Functional MRI lets us see how the brain is working or active when we ask people to perform a particular task. And diffusion MRI lets us look at these fibre pathways, the connecting wires between different brain regions, and shows us how strong they are in different individuals. So now I'm going to tell you a bit about some of the studies that have been performed by researchers looking at these kind of phenomena, looking at how learning and experience can shape the adult human brain. I'm sure many of you will remember this infamous work from London quite a few years ago now, about 10 years ago, from UCL, where they scanned, using MRI, they scanned a bunch of London cabbies in order to see if there was anything special about the brain of the London taxi driver. And what they found was this very specific part of the brain called the hippocampus was larger in taxi drivers than in regular people. And this is actually really interesting and compelling, because we know from decades worth of basic neuroscience that this area, the hippocampus, is particularly involved in spatial navigation, in navigating around space. And so obviously these taxi drivers who've had to navigate around the streets of London in many cases for decades, the idea is that maybe they've strengthened this special brain area which is involved in spatial navigation. However, if you were playing devil's advocate, you might say, well, hang on a minute, this is just a cross-sectional comparison between taxi drivers and other people. Maybe some people are born with a particularly large hippocampus, and that sort of predestines them to exploit that superior spatial navigation skill by, for example, becoming a taxi driver, and that you're not seeing an effect of that experience, you're seeing an innate difference between cabbies and non-cabbies. One bit of evidence they had in that paper which supports the idea that this is growth of the hippocampus triggered by this spatial navigation experience was that if they looked across their group of taxi drivers, they found that the people who'd been driving the taxi for the longest had the biggest hippocampus. The people who've been driving the taxi for the shortest period of time had a smaller hippocampus. So in other words, the number of years experience that they had of navigating around the streets of London seemed to correlate with the size of this brain structure, supporting this idea that maybe it was the experience itself which had changed their brains. But you can look at this question of experience-dependent brain change more directly if you study the same individuals over time. So take people, force them to undergo some experience and then scan their brains and test to see if that experience has changed their brain structure. And you can look at this not just looking at the grey matter of the brain, but also looking at the white matter. So we've been particularly interested in the white matter, the brain wiring, the pathways that connect different brain regions. You can think of these brain wires a bit like the wires in an electrical circuit. And if we were to ask the question, does white matter matter, we might intuitively think that it should matter. So that just as the quality of the electrical wires in your electrical circuit will influence how reliably your circuit works, so too you might imagine that the quality or the physical quality of the wires in your brain will influence how well your brain works, how well your brain is able to send messages between its different areas. So in other words, we might predict that the physical quality of the brain fibers will influence the functional quality of your brain, will influence the quality of signal transmission between different brain regions, how easily these electrical impulses can be sent from one area to another, and that in turn will influence your behaviour, because in the end of the day your behaviour depends on these electrical signals being passed between different areas in your brain. So this logic here would lead us to make certain predictions. It would lead us, for example, to predict that if people vary in the quality of their brain fibers, that might relate to variation in how good people are at performing tasks which depend on those brain pathways. So it will lead us to predict that as you vary the strength of brain connections, that will have consequences for behaviours and tasks which depend on those brain connections. And indeed we found that to be the case when we've tested it using experiments. So in this experiment, it's a very simple experiment in which we asked healthy young people in this case, to perform what we call a bimanual coordination task. So just performing a movement that involves your two hands. And this was the movement, so moving the fingers of your two hands in time with one another. And they were asked to do this in time with a metronome. Then the metronome gets faster and faster, and eventually all of us would be unable to keep time with the metronome. And so for every person, we can come up with a number which tells us how good or bad they are at coordinating movements across their two hands. Then if we take brain scans in those same individuals, we can ask the question, are there any pathways in the brain where the strength of those brain pathways varies with how good people are at coordinating their two hands? And we found a brain area where that was the case. So up on the top right there, again, this is a slice through the brain, a virtual slice through the brain, where in the middle of that brain image you see a little red blob. And that little red area there is a brain pathway where across individuals we found that the stronger that pathway was, the better people were at coordinating these movements between the two hands. So this is very interesting because it shows that even in healthy adults, there are variations in brain structure across people which have consequences for behaviour. So some of you will have a very well wired up version of this pathway, and you might be particularly good at this task if I were to test you on it. Others of you might have a more weakly wired up version of this pathway, and you might be less well able to perform this task. What's also interesting about this, though, is that it's a very specific spatial location which is showing this relationship. So it's not the case that all of the brain fibers show this relationship. It's not the case that some people have very well wired up brains and are therefore able to do well at whatever task I set them. But instead, what seems to be the case is that different brain pathways are responsible for different tasks. So you might have a weak pathway involved in coordinating your hands, but you might have a very strong pathway involved in language, for example, or music or some other function. So each of these different brain areas and brain pathways has developed somewhat independently from one another which fits our intuition that people are good at some things and bad at others. What this kind of work shows us is that people being good at some things and bad at other things also relates to how well wired up different areas of their brain are. So there are these relationships then between how well wired up people's brain are and how good they are at performing tasks. But again, we have this question of where does that variation come from? Are people just born with different brains and that makes them good at different things, good or bad at different things? Or does it partly depend on the experiences that you have throughout your life? There is evidence to suggest that experiences do shape brain wiring and brain structure. We heard about the taxi driver example. This is another example now looking at brain wiring in people who practiced the piano as children. So in this study, the experimenters found that certain brain pathways were stronger in pianists relative to non-pianists. Again, we don't know whether that's innate differences, that pianists were born with a different brain, or whether that's experience-dependent differences. Further evidence that those differences were due to experience resulted from the piano playing are shown in this relationship here. So what these graphs are showing us is that the amount of time people spent practicing these different tasks impacted on the strength of these brain pathways. So on the top right there, you see that variation in practice across the horizontal axis between individuals predicted variation in brain wiring. So the more time people spent practicing the piano between the ages of 12 and 16, the stronger those pathways highlighted in red on the brain image. On the bottom, you see that same relationship for a different brain pathway when we consider the amount of time people spent practicing over the age of 17. So the more time people spent practicing as adults or old adolescents, the stronger these yellow brain pathways that you see here. So what that shows us then is that brain pathways change with experience, and that at different stages during development, different brain pathways might be susceptible to change. So in young childhood, some brain pathways might be maturing and susceptible to change, whereas in adolescence, it might be different brain pathways that are now maturing and can be altered with experience. So given all these different observations when comparing special populations, we were interested in taking completely naive, non-expert people, training them in some new skill that they had no prior experience of, and then testing to see whether that changed their brain in any way. So we took people who had never juggled before, trained them to juggle over a 6-week period, scanned their brains before and after, and looked to see whether their brain changed as a result of that juggling. And what we found, surprisingly, was that it did change. So here again is a slice through the brain, a horizontal slice through the brain, and the red areas that you see at the back there are areas of grey matter that increased in size as a result of learning to juggle. In blue, you see brain pathways that strengthened where the connections were strengthened as a result of that training. So what this suggests then is that in healthy adults, learning a new skill over a relatively short period of 6 weeks is changing the brain. It's changing the size of brain areas involved in the task, and it's changing the wiring between different brain areas. In this case, the areas that have been involved are areas that we know are particularly involved in reaching and grasping to objects in the periphery of our visual space. So these are obviously functions that we would expect to be involved in the skill of juggling in particular. Okay, so those were just a few examples then of ways in which using these brain imaging techniques, researchers have been able to show that that experience shapes brain structure, and that learning new skills, even as an adult, changes your brain and changes the wiring of your brain. One thing that we've been particularly interested to do is to apply these types of brain scanning techniques to situations in which the brain is damaged, and in particular to stroke, to try and see whether we can gain information from brain scanning that we can then use to design new therapeutic interventions. So, as I'm sure many of you are aware, a stroke is a disruption to the brain's blood supply, which causes damage then to the area of the brain that was being fed by that blood supply. And stroke can affect people of any age, including children, but it is more common in people over the age of 55. And every stroke is different. So depending on the location of the stroke and the size of the stroke, the brain damage caused by the stroke can affect many different types of behaviour and activity. It can affect movement, language, senses, thinking. Every stroke is different. So here's just an example of one stroke here. So again, this is a horizontal slice through the brain, and the black area that you see on the right-hand side of that brain image is an area that's been affected by a stroke. So that's an area which has had a disturbed blood supply, which has then damaged all of the brain tissue in that area, and that area itself will not recover. Some people recover well after the immediate effects of a stroke, whereas some are left with long-term disabilities. And rehabilitation aims to help people cope with these disabilities and to relearn their skills. So the bit of the brain that's directly damaged by a stroke, in other words, the black bit on the right-hand side of that stroke, of that top image, that brain area will not recover, cannot recover. However, the rest of the brain that is still intact can, in many cases, compensate to some degree for the damage that's been caused. So there can be adaptation or compensation, or what we call plasticity, so changes in the way that the brain does things can happen to cope with the effects of that damage. There have been many studies using functional MRI to look at brain activity and how that changes when somebody's had a stroke. This is just one example that I'm going to talk you through now, where if you put somebody in the MRI scanner and did the experiment that we talked about earlier, so you have someone in the scanner and you ask them to move their hand, then relax, move their hand, then relax, we can highlight, as I showed you earlier, the areas of the brain that are involved, that are active when the person is moving their hand, the areas of the brain that are involved in movement. And you see the two brain images in that black box in the center there. Again, these are these horizontal slices through the brain. On the left-hand side, you see the pattern of activity that you would see in a healthy person performing that movement. Now, on the right-hand side, you see the pattern of activity that you would see in somebody who's had a stroke. So we could have a bit of a quiz now to see if can anybody, what do you think is the most striking difference between those two brain images, the healthy image and the stroke image? Can anyone spot any differences in the activation pattern between the healthy case and the stroke case? Any suggestions? The healthy's connected. That's
the other side
true.
is active in the stroke.
The other side is active in the stroke. Yeah, yeah. So the thing that's of particular note is that in the stroke case, you see activity on both halves of the brain, but on both the left and right-hand side of the brain. Whereas in the healthy, the vast majority of the activity is concentrated on one side of the brain. Now, as I'm sure many of you know, we know that one side of the brain controls the movements of the opposite side of the body. So if somebody's in the scanner just moving their right hand, let's say, we would expect to see activity pretty much concentrated in the left hemisphere of the brain, just as you see in that healthy case there. Whereas if somebody has had a stroke which affects the movements of their right hand, and they've somewhat recovered those abilities, so they are able to move their right hand, when they move that affected right hand, we would see this activity in the two halves of the brain, just like you see on the stroke side there. So it seems that in order to produce that recovered hand movement, both sides of the brain are now having to work. So it might be, for example, that the healthy hemisphere, the side that's not been affected by the stroke, is to some extent compensating for the effects of the stroke. So the good hemisphere can to some degree take over the functions of the stroke-affected hemisphere. But we also know that this pattern of activity in both hemispheres is associated with poor outcome in stroke patients. So the more the patient is able to look normal in their brain activity, the more the patient is able to concentrate that activity on one side of the brain, the better their hand function tends to be. Whereas we tend to see this activity in both hemispheres more commonly in patients who have recovered less well. And therefore we can think of this as something of an imbalance. So whereas in the healthy side, activity is very much tipped in favour of one hemisphere, in the stroke case, activity is much more evenly balanced between the two hemispheres, and that even balance between the two hemispheres is associated with bad recovery. And we therefore might suggest that one way of trying to boost recovery would be to try and reset that balance, to try and make the balance of activity in the stroke hemisphere more like what we see in the stroke person, more like what we see in a healthy person. So strategies to do that, therefore, might include either trying to increase activity in the stroke-affected hemisphere, which is underactive, or try and decrease activity in the healthy hemisphere, in the good hemisphere, if we think that it's overcompensating in a potentially negative way. So there are two ways in which we can either boost activity in the stroke-affected hemisphere or decrease activity in the healthy hemisphere. And conveniently for us, there are methods in neuroscience which can allow us to either increase or decrease activity in selectively targeted bits of the brain. So a method that we use is a type of brain stimulation method. It's something called transcranial direct current stimulation, or TDCS. It's being modelled here by a couple of my researchers. So it's a very, very low-tech type of brain stimulation. So it's non-invasive. There are two electrode pads, two rubber pads, which are placed against the person's head, and then a very small current, usually 1 milliamp, is passed between those two electrodes. And we know that if we locate one of those pads over the movement area of the brain and we pass the current in a particular direction, we know that has the effect of increasing activity in that movement area of the brain. And we can target this selectively at one side of the brain or the other side of the brain. So this is a method then which allows us to selectively increase activity, for example, in the stroke-affected hemisphere of a stroke patient. So lots of neuroscientists have spent a lot of time studying this type of brain stimulation in healthy people, and we've learnt quite a lot about the effects that it has on the brain. So we know that if you stimulate the movement part of the brain, you increase its excitability. We also know that it changes other features of the brain, and we think we can capitalise on those changes in order to use this type of stimulation to boost rehabilitation after stroke. And I'm going to talk you through the logic behind that. So the logic behind it is that motor training, so for example physiotherapy, different types of movement rehabilitation after stroke, just like learning a new motor skill, like juggling for example, relies on what we call brain plasticity. So learning or relearning rely on changing the brain in all of the ways that we saw illustrated earlier, and we call this brain plasticity. We know that this kind of brain stimulation has all sorts of effects on the brain which should boost the chances of plasticity or brain change occurring. We know, for example, that this kind of brain stimulation alters certain brain chemicals, and that the way in which it alters those brain chemicals makes it more likely that the brain will change. It also alters brain activity, as we assess using our functional MRI method. It alters brain activity in a way that makes it more likely that the brain will change. So we know that if we stimulate the brain in this way, it's more likely that the brain's going to change. So therefore, we might predict that if we can give our physiotherapy or our rehabilitation intervention while stimulating the brain in this way, then the normal brain changes that would occur as a result of that physiotherapy will be boosted because of our brain stimulation. So importantly, it's not that the stimulation itself is going to somehow magically cure the brain of stroke, but rather that if we add the brain stimulation onto an effective rehabilitation training therapy, then the brain stimulation will boost brain plasticity. It will increase the chance that the brain will change as a result of that physiotherapy training. So in the trial that we've been running within the Oxford BRC, we've taken chronic stroke patients, so patients who are many months, in many cases many years post-stroke, all of whom still have some difficulty with moving one of their hands, with one of their arms. And we've given them a very intensive motor training, movement training intervention. So they come into our lab every day for 2 weeks, and they do lots and lots of repetitive exercises. So here you see one of our volunteers performing. This is the exercise that they hate the most. They have to take clothes pegs from one place, unclip them and peg them onto another location. And it's very annoying because the clothes pegs keep pinging off. And it's very difficult. It's quite difficult for me to do this, but it's particularly difficult to do this if you've previously had a stroke and are still having trouble moving your hand. But nevertheless, they persist at this and they train and train over this 2-week period. So the hope is that that repetitive motor training across a whole range of different movement tasks will cause brain plasticity or brain change to occur, and that will improve the patient's movements as a result of that intervention. But what we're also doing is testing whether if we add brain stimulation to that training, if in half of our patients we stimulate their brain while they're doing this motor training, the theories would predict that because the brain stimulation is going to boost brain change or brain plasticity, those participants who've had brain stimulation paired with training should do even better than those people who've just done the training on its own. And indeed, that's what we're finding. So this trial is still in process, but our interim analysis is plotted here. So just to talk you through this graph, so we've got two lines, an orange line and a blue line. This is showing tests of a clinical score over time. And at the first time point, so the first point on the left is that at the baseline when the patients all start out on the study, then they're randomized to either receive this real brain stimulation or receive what we call a placebo stimulation. And so they're strapped up to the stimulator in exactly the same way. They don't know whether they're getting real or pretend stimulation. The experimenter doesn't know whether they're getting real or pretend stimulation. And they proceed in just the same way, but we know whether or not the stimulator is turned on. And then you see the follow-up time point. So immediately after that baseline, they get their 2 weeks of therapy, and then the next time point is immediately after that therapy. And what you can see, I don't know if I can point with this, is that at this time point, one thing that's really encouraging is that actually all of them have got significantly better. So whether or not they're receiving the brain stimulation, they've all done well with this intensive 2-week intervention. That in itself is actually very important because, as I said, many of these people might be many, many years post-stroke. Many of them will have assumed that there's not much scope for further recovery, and yet with this very, very intensive intervention, all of them have done significantly better after 2 weeks of intensive motor training. But then what's particularly interesting from our point of view is this difference between the orange line and the blue line. In other words, there seems to be this even more improvement in those people who've had their brain stimulated while they're undergoing the training. So this seems, in our mind, to support the idea that if you stimulate the brain during training, that boosts the chance of brain plasticity occurring. It boosts the effects of that training intervention, and encouragingly for us, that persists all the way out into this 3-month follow-up period. So we're following these people up over time, and that difference, the benefits are still there at 3 months, and the added effect of the brain stimulation is still there at 3 months. We're also doing lots of brain scans on the people participating in this trial to try and see if the brain is also changing alongside those improvements, and just one little result is shown there. Again, that's a slice through the brain, and the red areas that you see there are areas where the brain has increased in size in the people who've had this stimulation plus training. So it seems that the training plus stimulation is changing the brain, and that is having a positive effect on the patient's ability to move their hand. Okay, so then the final project that I want to tell you about is a relatively new project in which we're trying to test whether we can increase resilience to brain aging. So unfortunately, all of our brains will decline somewhat over time, and that decline starts relatively early. So from sort of, well, on these plots here, you can see from sort of middle age onwards, there's unfortunately a steady decline in our brain structure. So all of the size of our brain, the quality of those connections between our brain, the function of our brain, all these things slowly decline over time. But so that's a rather negative message, obviously, that things tend to deteriorate with age. But one much more positive message is that our lifestyles and our experiences and our activities can influence how steeply that decline occurs. So one example that we're particularly interested in is physical exercise, physical activity. There's a lot of good evidence out there now that the more active, the more physically active we are, the more we engage in physical exercise, the better our brains, the healthier our brains. Here's just one example. This is the hippocampus. This is the taxi driver area again, the hippocampus. So the hippocampus, as well as being particularly large in taxi drivers, it's also an area of the brain that's particularly vulnerable to the effects of aging and dementia. So in Alzheimer's disease, for example, this is an area that is very, very heavily impacted by Alzheimer's disease and tends to shrink in size, and that's associated with memory problems that people get with Alzheimer's disease and other types of dementia. However, it seems that this structure, the hippocampus, is also very sensitive to effects of physical exercise and activity. So this study here that came out of the States showed that if you took elderly people and enrolled them on an exercise program for a year, that had the effect of slowing the usual age-related shrinkage of the hippocampus. So on the graph on the right there, you see a plot of the size of the hippocampus for a control group in red who didn't undergo this physical exercise training, and an exercise group in blue who did undergo the training. And what you see in the control group in red is that over the year of the study, there's a steady decline in the size of their hippocampus, like we would expect in this group, that things decline over time with age. In the exercise group, it's very striking that they're not declining, and in fact, if anything, there's a slight increase in the size of that brain structure after this exercise intervention. And there's also evidence that that rescue of the brain is associated with improvements in thinking as well, and so it has a positive impact for memory and attention and other kinds of thinking properties. Here in Oxford, so we've started 2 large projects trying to look at this sort of lifestyle interventions for healthy aging, to encourage healthy aging and to try and offset or delay the onset of age-related cognitive decline and dementia. The web address that you see there is the sort of umbrella website which describes the different studies that we have ongoing. I have some leaflets at the front about one of the studies, which is a physical exercise study. So we're taking 2 different approaches to try and slow down age-related decline. One is to use physical exercise, physical activity, in which we're recruiting people to a 12-week exercise intervention where people who have relatively sedentary lifestyles currently are enrolled on a 3-month exercise training program, which is carried out at Oxford Brookes University, and we're looking at the effects that has on their brain, the effects that has on their memory and attention and other abilities. And on the right-hand side, our colleagues in Oxford are looking at what's called cognitive stimulation, so brain training, if you like, different types of exercises, not physical exercises, but mental exercises, so structured mental exercises, which again, there's increasing evidence that keeping the brain active, just like keeping the body active, seems to some extent protect us from the effects of advancing age. So to summarise then, we can use brain imaging to show that the brain changes its structure and function with learning and with recovery from damage. And by understanding how the brain changes in response to damage, we can begin to design new rehabilitation treatments. We hope that brain stimulation might be able to be used to enhance recovery and potentially also to accelerate learning in other contexts. And finally, changes in lifestyle can, we hope, boost resilience to brain ageing. So that's the end of the talk, and I'm more than happy to answer any questions, but thank you for listening.